The recording initiated, sir.
Okay. Yeah, we can start now.
Okay, sir. Good evening to one and all present here. I welcome you all for this webinar on programmable biology, fractal genomics, and precision medicine, informatics to rescue. I welcome the speaker, Mr. M.V. Ramanujam, sir, founder and chief science. Okay, sir. Now I request Dr. Prashant, founder and advisor of Bioclues, to welcome and give the opening remarks.
Yeah, thank you, please. Thank you for the kind words and introduction. Thank you so much, Shrecha, Mohan, sir, and Doctors Komathy and Sakthivel, for this wonderful organization. I had a great fortune in meeting Sri Ram in Bangalore a couple of times, and I really look forward for this wonderful talk. The talk is titled Programmable Biology, Fractal Genomics and Precision Medicine, Informatics to Rescue. This has been a kind of a need of the hour, thanks to the wonderful Nobel Prizes that have been won this year. Whilst we were celebrating the Nobel Week in the week of October 7th to October 14th, the first 5 Nobel Prizes which have been won largely dates back to biology, and if I can be brute honest, that goes back to something what we call it as data science and bioinformatics. So I'll probably give you a kind of a gist of it. Nobel Prize in Physiology and Medicine went to Victor Ambrose and Gary for discovery of non-coding RNAs in the form of microRNAs. Had it not been the microRNA databases and web servers that have been hosted by Gary, we would not have gotten this particular Nobel Prize. The second Nobel Prize for physics that went to, of course, the artificial intelligence, largely by and large with respect to how the AI and machine learning can be honored. That was how it went to the Hinton and his colleagues. And the third one went to Nobel Prize in Chemistry, that went to Baker's lab, where way back in early 2000, the computable RAM was probably used for discovery of umpteen proteins that were predicted using structural genomics approach at University of California, San Diego by Baker's lab. In fact, I was one among those particular first users where we dedicated our RAM, the unused RAM that was going all through the night, because we worked during the daytime from 8:30 a.m. to 8:30 p.m. So right from 8:30 p.m. to 8:30 a.m. The next day, our unused RAM would be remotely used by these particular computer predictions, where this particular inventions came to the limelight. The fourth one went to Kang for Nobel Prize in Literature from South Korea. So with a large amount of Ori repercussions that North Korea made on South Korea, Kang made several paraphrases, poetic paraphrases in Korean, which was later translated to Hindi, English, Tamil, Malayalam, and also in Han Chinese, which was read by so many people. But the beauty with her particular paraphrases was the data that she probably used, in paraphrasing those particular things. A large amount of, the mental health reporter that she used. Whilst writing these particular poetic paraphrases. The fifth Nobel Prize for Peace went to Idankyo, which is a wonderful organization based in Japan. So when the hydrogen or the nuclear bomb was bombed by Americas in Japan, there was a large amount of despair in Japanese, and just because Japanese would probably revolt back again, Idankyo organization came into a large amount of mental health data. They could ensure that peace and prosperity would really win by taking amount of all these particular health related data sets. So I can vehemently be very open in saying that we take immense pride as biologists and more as bioinformaticists or data scientists that the first 5 Nobel Prizes have largely gone towards development of data science or biology for that matter. And it's an immense pride for all of us that, this is being honored today. And I'm sure with this little introduction, Sri Ram's talk, on programmable biology, taking up precision medicine and informatics to rescue is being set up as a wonderful tone for this webinar series organized by CSI Chennai, IEEE, CS Madras, and ACM Chennai. I look forward to hearing your talk, sir, and thank you so much, for your gracious opening on this. And I also thank Shri H.R. Mohan, sir, for this wonderful and kind invitation. And on behalf of Gyaneshwar Chaubey and Bioclues, unfortunately Gyan couldn't make it. He's sick, organizing these wonderful INBIC series the last week, and he conveys his best wishes to this wonderful team. Thank you so much, and I look forward. Namaskaram. Yeah.
Thank you.
Thank you, sir. Thank you for your wonderful welcome speech. Now I request Mr. H.R. Mohan, Chair, Events, IEEE CS Madras, to introduce the speaker.
Greetings and good evening to one and all. So let me once again welcome on behalf of the organizing units IEEE Computer Society, CSI, ACM and Bioclues. A warm welcome to Mr. Ramanujam, the speaker of today's webinar, the members and my colleagues from the organizing units, and the participants who joined the Google meeting platform, and those watching the YouTube streaming of this webinar. For the benefit of our members, let me provide a brief on Bioclues, the largest group of 9,000 plus professional students, faculty and researchers in biomedics and allied areas in India. It has been functioning very efficiently in imparting and exchanging knowledge among the stakeholders for the last 20 years. We are fortunate to have Dr. Prasant, the founder of this Bioclues, to join us this meeting and do the welcome address and give an opening remark for this webinar. I would like to encourage our members from CSE and IT areas to get affiliated with Bioclues and be a part in the advances in life sciences research. Coming to today's presentation, the marriage of life sciences and computing in the form of bioinformatics, computational biology, etc., has brought out a lot of innovations and advances in medical and biotechnology areas. Large-scale collaborative projects with groups of biologists and computer scientists are happening globally, with each other learning from others. Now, we organize events of computer applications in life sciences. One of the earlier most popular webinar we had organized was applications of machine learning and deep learning in life sciences by Dr. Jayaraman, a former NCL scientist and the current professor at Flame University. Today's presentation is going to give some basics of groundbreaking technique, genome editing and engineering using CRISPR and Cas9, which led the emergence of new informatics-based industry genome engineering. In this presentation, principles of governing the fractal genome will be discussed, and the importance of understanding mathematical underpinnings of the code also will be highlighted. India's expertise in mathematics, informatics, and computation, coupled with advances in genomic informatics, could significantly enhance its involvement in the global genomics and genome engineering sector. All these will be deliberated in today's presentation. Coming to the introduction of today's speaker, it was recently I got connected in LinkedIn while I was looking for some references in bioinformatics and related subjects. Mr. Ram's extensive experience in industry, academia, and research made me to request for a webinar presentation, for which he had readily agreed. Mr. Ram, popularly known as, Mr. Ramanujam, popularly known as Ram, is currently the founder and chief science feature officer of Prodigious, Quant-Epigen. Ram began his studies in biology in 1982 and has since been educated, trained, delivered numerous keynotes, and collaborated with major global organizations worldwide. He has advised and mentored numerous startups in modern biology and is currently an honorary member of the Science Advisory Board for many emerging companies. With over 3 decades of global corporate experience, Mr. Ram was included in the Webster's Timeline Series edition, recognized for his pioneering discovery. And for the first time in the world, in this RZ Diagnostics, a double-labeled RNA exhibits quenching of the first time globally. He participated in the development of neurotropics and neurotropic factors for clinical application and was part of a neurobiology major in the US, being a pioneer in the application of fractal genomics to address computing economy. With the 3 decades of expertise in discovery biology, genomics, and computational sciences, Mr. Ram is currently focused on advanced molecular cell medicines and therapeutics and molecular immunology, including immunotherapies and RNA sciences. He has collaborations worldwide. He is also the founder of Global Fractal Alliance. Mr. Ram is highly regarded corporate researcher, recipient of the Lifetime Achievement Award from Non-Profit Research Center in India in 2017, Inspiring Bio-Entrepreneur in 2023 in Mumbai at the Bio Enclave. These are some examples of his research recognition. With this brief introduction, let me now request Mr. Ram to enlighten the participants on the role of informatics in programmable biology, fractal genomics, and precision medicine. Over to Mr. Ram.
Thank you. I first want to take the chance and the first chance in thanking all the organizers of this particular event. And I will straight away go to the slides and just give me 1 second. And I think I should be able to, I'm able to load that for you. And so that you will be able to look at that. And so first, I want to make a disclaimer that this is not a seminar on genome editing, because that is a well-beaten down track right now, because it's more than 10 years that people are aware of this genome editing technologies. And what I am planning to do today is work towards fractal genomics. People ask me, what is fractal genomics? Because we know fractal, we know genomics. What is fractal genomics? And personal medicine, it is again beaten down track, and precision medicine, this has been talked about for a couple of decades. How does informatics, and as Dr. Prash talked about, on all this design of new proteins that has come alive now, and also I saw some AI-based approaches for the that you are already aware of, AlphaFold 2, 3, and also for the RNA, some of the AI-based technology. So my talk in the first 10 minutes or so, it will be more on introduction, And I start with Edwin Chargav's this particular comment that he made. I started from the conviction that if different DNA species exhibited different biological activities, there should also exist chemically demonstrable differences between deoxyribonucleic acid. And I can tell you a story. He was a professor at Columbia University, and for his discoveries, and I regard him very highly of his science, without whom we would have not gotten to the level of discovery of the DNA in 1953. So the ratio. So this is, to begin with, to tell you, because there are a lot of students from across the informatics side, I want to go through a little bit of introduction here. This is Jim Watson, and he actually, along with Renato Del Becco, way back in the 80s, started the Human Genome Project, and went on with the DOE presence, and DOE's help the whole world knows about the cottage style human genome sequencing with the Sanger sequencing that was predominantly done. And I will take you through Leroy Hood and Francis Collins. Francis Collins being the leader of the Human Genome Project, and I'm coming to the point of Craig Venter also. So Leroy Hood is the person who worked on the sequencing machines, and collaboration went on, and so some critical comments were made. Like, we often seem to be patching holes and building on top of old architecture instead of tearing down and remodeling the right way. And I found that pretty interesting from a biomedical informaticist and a bioinformaticist, Nephi Walton, from the University of School of Medicine at Washington. So coming to the point of Craig Venter, what is his contribution? He hastened that he speeded up the whole process of human genome sciences and human genome sequencing. And what took more than 20 years, he could do it within 3-4 years. In 2003, we had a whole human genome draft and the live code. And at that time, we all know that the most disruptive technology, and I was reading a paper in Science in 1996, sitting in a library, and I realized the bioinformatics is going to be the future. And this is one of the most disruptive technology ever to be to come on board. So the Eric Schott, the director of the multi-scale technologies, multi-scale omics, rather, Eric Schott is Mount Sinai, director of Mount Sinai, and his contributions are immense. And I'm just showing you because some of the key contributors and key players, movers and shakers of this area, how they did this. And I come to Dr. Pelianis. It's a very interesting person, and I discovered him in California way back in 2010, and then I collaborated with him extensively along with his colleagues, Jean-Claude Perez, and other colleagues, Dr. Sergei Petakov in Russia, and as well as Dr. Andras Pelianis, Dr. Jean-Claude Perez, and Professor Thirajan Govindarajan in Hyderabad, who happens to be the grandson, and I mean, his cousins, Srinivasa Ramanujan's cousin's grandson, actually. So this is highly mathematical. The mathematical rigor is well known. And Dr. Pelianis is a triple doctorate from Hungary. He is a student of Janos Setagodai, and he's a neurobiologist and a genomic scientist. So with his help, we actually brought him down to India, and he is, you can see his picture showing the Padmanabha Swami temple in Trivandrum, and we collaborated. All of this collaboration went on. It's not just my contribution alone. I took active interest in how this can be commercialized for the Indian hemisphere and how we can actually go about commercialization. And that is where the approach that we have taken, genome engineering, synthetic biology, programmable biology, approaches for the statistical diagnosis, probabilistic prognosis, and precision medicine. And I will come to that a little bit for the sake of the students and the brevity of the topic, and I'm giving you a little bit ambit of introductions. Please bear with me. In October 20th, on October 28, 2008, a Google talk was given by my collaborator. Is IT ready for the dreaded DNA data deluge? And today that nobody is willing to pay for bioinformatics. A lot of people are willing to pay for sequencing. It is assumed after the sequencing, bioinformatics is also allied to that. And so many service companies that exist in India charge accordingly. And at that time in 2008, the bioinformatics was bottlenecked. And data analysis, the data sciences as such was just coming up. in 2008, and you see my collaborator along with James Watson in India showing his fractal temple, and he is along with them. So let's just give you a few pictures. So now I'm getting to the real crux of the seminar and what is happening with the mathematics and what is the mother of all sciences, and courtesy of the Fractal Labs initiative, public domain literature, all of them quoted here. The main point here of this particular slide is Benoit Mandelbrot, who introduced the fractal geometry. And he understood and he has shown the fractal geometry of nature. The clouds are not spheres, mountains are not cones, coastlines are not circles, and the bark is not smooth and not as lightning traveled in a straight line. So he arrived at, based upon some of the works that were done previously, and he introduced the fractal geometry, and he is French-American, worked in IBM. And fortunately, my collaborator, Jean-Claude Perez, is also a collaborator with Benoit Mandelbrot. And Benoit Mandelbrot is a tall figure in fractal work. And so the mathematics, I would say, infiltrated biology way back in 1600s by René Descartes and people like top biologists at the time who were doing biology. They infiltrated mathematics into biology. So this is not new. Informatics would become part of biology, and genomics is, after all, informatics. And we understand that very well right now. Genomics is informatics, and even we thought in 15 years back that many of the pharmaceutical companies will be IT-driven pharmaceutical companies, informatics-driven pharmaceutical companies. And that's exactly what happened. AI took over, and AI-DD, AI-based drug discovery, development. Now more than about, I would say, billions of dollars have been poured in the AI-DD. I'm coming to the point at a later slides. I'll be explaining them a little bit much further. So I'm showing you fractal geometry and fractal work is not new to India. We know this. We have done this. We were very good at it. Our Gopras, as we explained, the Mother Meenakshi temple, and as well as this Khajuraho temple and other Indian temples, self-similarity. So what is a fractal? Fractal is a self-similar repetition. And the self-similarity and 3D fractal is well established in Indian science as well. So Dr. Andros Pelionis came along and visited all the temples in Srirangapatna and as well as in Padmanabha Swami temple. It looked to me that they knew more about our fractal temples than I did. And then I went on researching on them. And that's when I realized the Russian dolls, these are fractal dolls. These are all called Matryoshka dolls. And I'm showing one more figure here as the Russian dolls. So not until Francis Collins came to Bangalore and said that, what strengths do you have in Indian institutions? In the Indian institutions, traditionally, we are very good in mathematics, very good in statistics. We were known for a lot of credible discoveries in mathematics and statistics, and as well as in chemistry. And India's greatest strength now is IT and computational biology. I will tell you where we have gone right and where we have gone wrong also, in terms of this is not a services industry. This bioinformatics, when it arrived in Indian spectrum, people thought it is more of a services spectrum. It's not. It is more academic. It is more depth information. It is a biology driven, and you need to know both coins, both sides of the coin, informatics and biology. It is extremely domain intensive, and that's where India is on the right track. But still, we have a long way to go, because this is something that Francis Collins told, that India is inherently quite strong in mathematics and informatics and statistics. These are the tools that is required, the tools that are required along with biology. So in 2007, the story was different. Genome informatics, and 98.7% of the genome is a junk, was not realized at that point. And then we knew that it is this part of the genome is anything but junk. And therefore, if you just calculate 3.2 10 to the power 9, 7 billion, now 8 billion people into 100 trillion cells, and if everyone has to be sequenced in the human earth, and every other species have to be sequenced, and we will have a huge amounts of data, and analyzing the data will require scores of people. So it will become more like a cottage industry. And there was one answer when somebody asked Francis Collins, who can be given a Nobel Prize for genomics. the genome sequencing. He said, give it to thousands of people who worked on this. So giving that point and getting into this, some of the platforms, and when I was discussing with the organizers, they realized that some of the from academic side, in the informatics side, and then you need to know a little bit about sequencing. There are lots of platforms for sequencing. This is a Illumina platform. I won't go through all the detail. This is all beaten down track. Everybody knows about it. And people are very comfortable about it. They're knowing this. One is Illumina platform for sequencing, PacBio platform for sequencing, both for whole genome sequencing and as well as exomics and as well as your microbiome, whatever you want to do. This is Promethean, ONT, Oxford Nanopore kind of technologies. And this is another platform, MGI and other platforms are there at the moment. So with that introduction, I said earlier that René Descartes in 1629 infiltrated biology, at least since René Descartes. And he is also a philosopher, by the way. And in 20th century, the Ludwig von Bartholomew in 1934 introduced System X, cybernetics, and informatics. These are very important terms. And we, along with Professor Ethirajan Govindarajan, conduct these conferences very regularly, the international conferences in System X, cybernetics, and informatics. So the cybernetics, which was brought about, is about a feedback loop, a concept of the feedback, 1948. And cybernetics would include engineering, linear dynamics, system control, computer science, biology, neuroscience, philosophy. And Hopfield himself, this year's Nobel laureate, he is an expert in neuroscience. So, I can tell you that until Schrödinger, What is Life book that he wrote, and he actually gave a lot of lead for Jim Watson before the DNA discovery was done. Aperiodic, he said, is aperiodic. Hydrogen bonding over an aperiodic crystal. Crystal was later found wrapped around the double-helix core of the DNA. And so the math in the genomics is unavoidable. Unavoidable. Again, I'm saying, if you do not know the mathematics, the mathematical underpinning, the pristine language of the genome governed by the fractal work, you probably did not get to the essence of the genomics yet. So mathematically, the Hilbert curve. Hilbert curve is David Hilbert, 1891, German mathematician. Lindenmayer fractal is all known, and L-string systems, and Lindenmayer's system is known actually. The geometry of the nature was done by Mandelbrot. And later papers published by Eric Lander and said that, Mr. President, the genome is a fractal. So 2009 Science cover page establishes the genome is a fractal. So I don't have to beat that again and again. And so this is exactly as you read. This is if you have a diamond and if you have imperfections in a diamond, you can go ahead and correct it. You can actually do a structural functional syndromes of the pathology can manifest as a fractal defect. And these defects manifest itself. And then once you are able to correct that fractal defect, it can be a copy number variation, fractal defect. It can be structural variations. It can be smaller mutations, n number of mutations. So now we have coming to the programmable biology point of view right now is that now you have the CRISPR system and a Cas9 system. You are able to edit, you are able to prime edit, base edit, you are able to write, you are able to delete and add sequences. So such things we are able to do now. It's not how we can do it. It is what is that we are doing, where to edit. How to edit is a rather question that is obvious. But do we understand the mathematical underpinnings of the genome to point out where the fractal defects occur in your human genome? So what to edit is a question that you may want to keep in mind. So the concepts go on. They said, Craig Venter said that our concepts of genome regulation is unsophisticated, frighteningly unsophisticated. The reason he said that usually people think about genomics, but one has to think about also epigenome. And that is one of the reasons my company's name is Prodigious Quant-Epigen, pointing to the fact epigenome, quantitative, quantum computing that is going to be involved in that, and prodigious and copious amounts of data that is available before us, and even with the emergence of the AI, DD, and the AI now, the recent paper that came out in Science, EVO. So I will go through that a little bit later when I come to that point. So embracing the generalization, what is exactly is these are concepts that are a little bit difficult. I don't expect everybody to understand immediately. The covariant, contravariant functors of the eigenstates is a mathematical description where we now describe the covariant protein signaling RNA, non-coding RNA, and as well as RNA vectors that are executory, contravariant. So this covariant, contravariant functors of the eigenstates are very important because that is how we are able to delineate the coding region and non-coding region. We are able to say that because what is happening here, these are usually symmetrical regions that are there in the genome, and as well as symmetry is well proven, and this repeats that are also well proven. They are like palindromes, they are like repeats, they are like inverted repeats as well as direct repeats that occur. So the last point that I'm making here is the zip of Mandelbrot parabolic fractal distribution law of the frequency and ranking diagram. What that means is very simple. I will explain it to you because this is a little bit complex actually. Zip-off is actually this is in the language, in the grammar, it is actually being given. And Mandelbrot parabolic fractal distribution is nothing but I will just show you. I'll go through that diagram immediately because I have said that in one more time. This zip-off function, I have it. This is the zip-off on the right hand side that you are seeing is the zip-off Mandelbrot parabolic fractal distribution. So when such a parabolic curve occurs, this can be shown in a smaller organism. The compute power that is required to do such kind of calculation is immense. And therefore, for a human genome, such a parabolic curve has not been shown so far. So this is the zip-off Mandelbrot parabolic fractal distribution curve. I will come to you again. I am starting to explain this concept because there are lots of concepts that is coming through. So this is a picture of that where it shows the tumor development and fractal structure, fractal distribution, and we call it as a fractal dimension. And it can be calculated. The fractal dimension can be calculated. The irregularly shaped tumor vasculature. So the fractal pathologically can also be the fractal structures can be identified. So this is all histopathology type of work. And here you are showing the intratumor heterogeneity and allelic frequency, and you are seeing all these are fractal structures that you see here. Okay, so far I have shown you some of the examples. For the genome is a fractal. Then I've shown you how the genome can be edited. I've told you about that. I've not shown you, but I've told you about that. And the precision medicine, I'm coming to that point. So these are some of the papers. So I'll quickly go through. These are shown in the cancer research and fractal cancer. If you go to the Google and just say fractal cancer, you will get enormous number of hits. If you say fractal genomics, you will get enormous number of hits. So this is a well-known area, and commercialization of this in India, nobody has done. Oncological mathematics, briefly to tell you, this is a new field. People are working on it. And this is about the fundamentals of the tumor behavior. People can study loss of genetic stability, tumor growth, immunological identity, so forth and so on. Okay, I have a little bit more slides just to make the brevity a little bit easy, and also to go through the slides as we go on. The story gets explained. The genome is a fractal, and using math in biology, this person Gabriele Losa is a Switzerland person, and he's a researcher, and he is well established. And in neuroscience, he is a highly notable person. Actually, he has done a lot of work. So one more example, fractal dimension. As I mentioned to you, fractal dimension can be calculated as a novel measure for the prostate cancer grading. So initially I told you about the diagnostics that can be done with this kind of fractal dimension. Next I told you about probabilistic prognosis, and third I told you about the precision medicine. I'll come to the precision medicine a little bit later, but as far as the complexity is concerned, you see the complexity. Genome probably sits on the top. The complexity is very high. And other things, when informatic way, if you have to describe the complexity, the genome, the smartest, but most user unfriendly secret of the universe. So this only two lines I want to show here. A massive computational problem looms in interpreting this data, told by David Osler. the professor of genomics in the UCA, University of California, Santa Barbara, I think, if I am right. So going further, the patent protection was required for this, and the patent was already filed in the US, and it was approved, and also papers were published. So these are all well-established concepts, and the leaders in the field are collaborators. And you see the fractal genome, fractal folding of the genome. This was published by Eric Lander in Science magazine. And go on, I'm saying, Madam President, the genome is a fractal, and you see the 3D structure of that. And obviously, you're able to see the globule structure, which was done by Aries Lieberman in the University of Houston. And he shows very clearly he has done a lot more work. They use techniques like high C and this H and small I and -C, high C techniques to arrive at this kind of 3D globular nature. Representation of the L system, as I explained to you, these systems are Hilbert curve. And I'm coming to the point why I am showing this L system again and again. And here again, the L system is shown and fractal governed by that. And the fractality of the genome, now it is. The precision medicine point is how you can actually eliminate this fractal defect, how you can intact fractality, which is compromised, is causing the diseases like cancer, how you can eliminate such a thing and then arrive at a precision medicine. And here I am showing first stage, stem cell stage, shark stage, frog stage. This is the proprietary knowledge, proprietary fractal approach, fractal gene approach done by Professor Andres Pelionis. And the equation that he suggests is Z is equal to Z squared plus C. And if, to simplify the concept, if this is anomalous, then it can lead to the fractal lung cancer tissue, actually. So the targeted fractal defect for the diagnosis by a ranking therapy is possible. Therefore, as you see from the simple template to a complex human stage, how the fractal is building up. So having shown that, this is another picture, same proprietary information, Pelionis proprietary fractal approach. He has it, and he is holding the patent for that. And I am well aware of that, what's know-how in that. But what exactly the issue here is the power of the fractal algorithm, both to separate harmless parametric diversity of individual genomes from syntax problems. So that is a point. So C is a constant here, but the syntax, the disruption of the syntax leads to the fractal structure, and therefore it leads to the gross fractal growth, such as cancer. And that's the point here. So if you have any questions in the future, in the question answer session, I'll be happy to answer you. Some of these things I'm going a little bit faster. I have a number of slides, and this is again showing the fractal brain and fractal genome and self-similarity and recursivity. And this point of recursivity is also very important because, here you are seeing a DNA, and you see the fractal growth as you go on. And what happens when the fractal growth happens, it puts this is the if the DNA is methylation, is interfered by epigenomic or hologenomic unregulated cancerous growth. That is the result of it, actually. So what happens in this situation, it puts a cookie in that DNA, and the fractal growth keeps on growing. When the fractal growth stops, then it will stop putting the cookie on the DNA. So that is how the epigenome, that is a recursive genome function, genomic and epigenomic iteration to regulatory DNA synthesis of hologenomics. This was suggested way back in 2008. So this is not new. A lot of papers have been published on it. And it is in a seminar, is IT ready for the dreaded DNA data deluge? In that seminar, it was presented. So I'm just borrowed that. And again, you are showing this laser that is pointing out your spaces here. So if you take this as a particular DNA, and if the DNA is a 3D structure, let us say about 6 meters of DNA, it is in a 2 micron space, and it is in a coiled structure with the histones, and the DNA is coiled and all that. Then you have a transparency. So when this transparency is clogged, that means the intact fractality is compromised. So when it is clogged, then what happens? Your transcription of the proteins out of this particular DNA, it gets hindered because one, it is clogging transparency, disrupts the functional proximity. Therefore, several transcriptional aberrations may happen. So this is a little bit more complex than that because we have to do a lot of work on particularly diagnostics as well as in the clinic to prove all these concepts. sorry, at the moment is just an academic exercise. At the moment, I will show you this. I've already shown you the parabolic curve. And here is an example of how the lung cancer is already shown, fractal structure is shown. And I'm also showing where the genome editing is possibly getting done. This is Benoit Mandelbrot and the Gobra of Sri Ranganatha Temple. So it's kind of what Dr. Andras Pelionis was thinking, and also he discussed with me several times, is to come up with a DNA report in an iPhone, actually. So you can have this diagnostics done and the doctor can actually go through these diagnostic tests and then immediately give you the report. So it is called a research kit, a DNA kind of report. And his historic discovery is that when James Watson and Crick, when he presented in a paper that DNA goes to RNA to protein and it never recurses. And so the double axiom, one is the junk DNA and the second one is the removal of the two dogmas. One of this is DNA to RNA to protein. It's not unidirectional, as he clearly showed, Crick showed in a paper with the writing, in a clear writing, it is unidirectional. But it is actually shown long time back in 70s that David Baltimore and Howard Temin have shown that this is not true. And then again, now we come to the 2000s and post 2000s. We know that for sure the transcription factors are binding, and even before that we have known transcription factors are binding DNA, and this can be recursive. This is a recursive PRGF, the principle of recursive genome function. So having said that, you can see this colored repeat C, G, C, G, G, C. And again, you are seeing that C, C, G, C, C. So these kind of repeat expansion, they call in ataxia and cerebrospinocerebellar ataxia and spinocerebellar Friedreich ataxia, or even the 40 different repeat expansion diseases. We are able to see this kind of repeat expansions. These are called GAA repeat or GCC repeat, or this kind of repeats come in and they are all mutated all over. Those mutations occur all over the genome of these kind of diseases, and they are all fractal defects. And in fact, they are fractal defects and they have to be neutralized. And one of the idea is how to neutralize those fractal defects. So this is an independent experimental proof of the concept fractal defect causes cancer and the structural variation in the cancer genome. It has all been published. It has been more than a decade having known all this fact. So this is how the industrialization we have working on some of the concepts. I won't be able to reveal everything to you, but at least I'll be able to tell you there will be full genome sequencing, genome analytics, and then you can do 3 ticks. One is a pharma, and one is a cancer center, and another one is a hospital. So you can measure the drug efficacy by DNA fractal defects before and after the treatment. And then you can develop with a genome-based treatment based on the efficacy with the DNA. And third one is the DNA fractal defect pattern determines best matching therapy. So I can tell you in chemotherapy, there are thousands of agents. And a doctor, whenever he gives a trial and error basis. Trial and error basis alone. If, for example, if we have a methodology by the whole genome sequencing and we are able to tell a particular precision chemotherapeutic agent, there is nothing like it. So this is one of those attempting such efforts. Again, I showed you about the diamond, and I can show you there is an imperfection in the diamond, and you can fill it up, fracture filling. And this is similar to the fractal defect neutralization. One can identify the fractal defects, structural variants, copy number variants, copy number alteration, or copy number variation. Structural variants can be very large. It can be many thousands of sequences. Nucleotides. In fact, so such large deletions and additions and all kinds of structural variants, they all form fractal defects. So this is the closing the gap, cancer gap, and I'm coming to the point of the programmable biology. Now, in programmable biology, the Ginkgo works in companies like that which are worked on that and working on that, and other companies are also following. Now there is a company called in CRISPR Therapeutics. They have come up with a therapy for that. All this is fine. I mean, it works in the case of a single nucleotide or not multiplexed situation. But in the case of multiplex situation, we still don't know. There are a lot of studies have to be done. And therefore, this particular child has been cured. That is a good news. and you are able to remove a particular sequence. You can do all kinds of things: prime editing, base editing, genome writing, genome addition of sequences, deletion of sequences, all that. But that's where we published the paper: Genome Editing, a Novel Business Opportunity for India as a BRICS country to excel in global genomic enterprise. What we say in this paper is three concepts. One, the genome engineering, the mathematical underpinning, the spell checker. It is like a spell checker on the genome. So if you are able to have an algorithm, proprietary algorithm, a proprietary informatics approach to go through the genome and tell exactly where to edit. So that is a grammatical error detection. So you are detecting that error, and then through synthetic biology experiment, the CRISPR-Cas9, or you are talking about Cas10A, or other newer versions of the Cas from Feng Zhang lab or Doudna's lab that where people are working on. And if you have such methodologies, and you can combine that in a way with this fractal genomic efforts, and therefore you will be able to arrive at an enterprise. So this is what we published. And we also presented it. And so this is, again, the same thing. Precision medicine, I want to conclude there as saying it is the fractal defect, the CNV, CNA, ought to be neutralized, not by trial and error methods of chemotherapy, but by genome-guided therapy, the GGT. So this has been going on in the Mayo Clinic, and there is a clinical trial that happened in the Mayo Clinic. And so what is that modern gene editors don't know what to edit and how they set out to edit. So knowing what to edit is a prerequisite requirement for DNA and RNA editing and for the shooting the messenger. So the CRISPR method, base editing, prime editing with the epigenome editing, along with RNA editing, need to assess the fractal defects. So that is where I come to the point. Now it looks very astonishing era in the DNA sciences and RNA sciences, particularly shooting the messenger and RNA. And finally, I want to mention it to you. I'm very interested in the immunoscience part of it. And that is one of my company's prime focus right now, is that in the cell therapy and genetic therapy, particularly in the cell therapy, in terms of T-cell engineering, and this CRISPR-based methods are frequently used. And therefore, knowing the fractal, when you engineer a T-cell, knowing and improving the T-cell engineering methodologies to improve the tumor immunogenicity when you are attempting against a B-cell antigen, those are the newer concepts. One we are working on. We are in the very preliminary stages at the moment. And this is the EVO concept that was published in the Science, because I was told when I was talking to the organizers that some of you will be very interested in the AI-based, ML-based methodologies, NLP-based methodologies. So this is something you can check out. This is a new paper on the Science cover magazine, and people are talking about it. And with that, I just want to acknowledge my professors, Andres Peliones, E.G. Rajan, Sergei Petakov, Jean-Claude Perez. All of them are collaborators and have been mainstream helping me out on all these technologies. And my company is in a stealth mode and is also tarmac mode. It is yet to achieve any funding, and we are working on these closely with our collaborators. Thank you.
Thank you, sir. Thank you for your detailed presentation. So we have some few questions from the chat box and also at the time of registration. First, let me pose the question which has been posted in the chat box. Rather than sequencing DNA and analyzing it, is analyzing the fractal structure of DNA cost effective, compute effective?
Can you repeat the question? Rather than sequencing the DNA?
Rather than sequencing DNA and analyzing it, is analyzing the fractal structure of DNA cost effective or compute effective?
Yeah, but without the sequence, for example, I think I should mention it to you. We need the genomes of the people who don't have cancer disease or any other disease. So we have to have the sequences. Unless we do the whole genome sequencing, we will not be able to analyze the fractal defects. That is very simple. So of course, you are talking about the mutant. Sorry, there is a power cut here. So you are not able to see. Just a moment. Can you give me 1 second? Actually, I will turn on the generator.
Yes, sir. Sure, sir.
I'm sorry. Sometimes it happens.
Yes.
Yeah. So, you know what happens? You have people who don't have any cancer, who are normal patients, normal volunteers or healthy volunteers. We have to have the sequence of their DNA, whole genome sequencing. And then we have to have the sequencing of the cancer patients as well. So the comparison and finding out the fractal defects in the cancerous patients are the key for doing such analysis. So we can take already sequenced lung cancer genome or any other stage 1, 2, 3, 4, any stage of the lung cancer or any other cancerous type cell, and we will be able to find the fractal defects. But without sequencing, without data, we can't do anything.
Thank you, sir. Next question. How can college students get into the field of fractal genomics? What are the skills and knowledge they must have? How will fractal genomics and bioinformatics affect the food industry?
Yeah, so I don't know about the concept of the food industry. I'm working on in the kind of healthcare. And the point is that to get into this kind of area, you need a interdisciplinary. You need to be a biologist knowing informatics, you need to know mathematics, you need to know chemistry, physics, mathematics, and biology. So it is really interdisciplinary nature of the science itself, which is what many people are doing right now in the world.
Thank you, sir. Next question. How can this webinar can be used in antimicrobial resistance research?
This is nothing to do with antimicrobial resistance. That is a different issue. People are coming up with some antibiotics and other molecules, or even you can come up with RNA target for small molecules. That is a different research, actually. This is nothing to do with it. We are not working on antimicrobial research at all, in fact, with this, actually.
Okay,
we are focused on cancer, in fact.
Okay, sir. Sir, informatics a boon or a ban in future years? Is there a cure for heritable diseases?
Yeah, there is a cure for heritable diseases. It is in the preliminary genetic medicines. People are working on single nucleotide or small sequences, whether we can replace them and we can have genetic medicines. The point, along with this, first question, please. What was that first question?
Whether it is a ban or boon.
Okay. Without informatics, we wouldn't be able to do all this. It is a boon, definitely. I even told you in the beginning that IT-driven pharma, that was the concept in the 2000s. So whether IT will even shorten the life cycle of the drug discovery, that is exactly what people are doing now in AIDDD. That is called AI, Drug Development and Discovery and Development. But what is the ultimate? Is it 5% of improvement or 10% of improvement? You are able to go to the stage of the preclinical trial by very fast time period, but we cannot avoid the preclinical and as well as clinical trials. So we have to go through the clinical trials. We have to understand how this molecule works in a human body. Therefore, ultimately, the success of the all informatics-driven approaches will depend on clinical trial.
Thank you, sir. Next question. How you think genomics can make remarkable discovery when compared to other omics?
Yeah, it was told that previously we knew about the 500 targets. Targets are the target discovery. And upon genomics arrival, the genome sequences arrival, we knew people said 5,000 targets. So what happens? Too many, plenty of targets are available. But at the time, target validation, then optimization procedures, then this are all multi-million-dollar enterprise. You know, if you have a target and you have to discover a particular target and validate a target against a particular drug, whether it is a small molecule or a large molecule, you are still talking about $250-300 million investment. It's not academic exercise. This, my uncle works in Texas A&M University. Once I told him that you need 10 Texas A&M universities. So that's what makes Pfizer. It is not one university or a single lab which can do drug discovery. It is like a relay running, then you have discovery, you have development, you have preclinical, you have clinical development, you have then phase 4. So all this is a very expensive game. And St. Gallen's report came out that 95% of the drugs that are discovered in the world are failing. Only 5% to 7% are succeeding. So who's going to pay for all the failures? So they reported $6.16 billion to bring up the drug into the market. So if that is the case, if we say a few billion, that may be the other calculation. But even if you say $2.5 to $3 billion, now we don't have such resources in India. So therefore, we will be better off by doing our own strengths: informatics, mathematics, statistics, computer science, and offering these multinational pharma and pharmaceutical companies our core inherent strengths, and therefore help the drug discovery. So that is exactly where we are right now.
Thank you, sir. Next question. How accurate and trustworthy can be AI and IOE sensors in the precision medicine? Next, the same person has asked one more question. Can AI-based technology really assist today's medical world in real time precisely?
Yeah, it's all good. Boils down to data, and again the availability of the data and how you are training these networks actually. So therefore, the precision will be dependent on the type of data that you are actually training that particular network. And the second point is whether you have the AI-based drug discovery come of age. I don't think so. It has not come of age. It is in the preliminary stages. And second point about the diagnostics AI-driven. Yeah, there could be some very good improvements in AI-driven diagnostics. So there could be biosensors, then AI-driven, then this kind of people have been working on fuzzy logic very long time back, those kind of things. But right now, I think AI has taken up AI ML methods. And see, moreover, the data science part of it is much more helped with the AI-based approaches rather than for the basic biology itself, because experimentation is the crux of the drug discovery and development.
Yes, sir. Thank you. Next question. As a student, I would like to know, it's been asked by Aishwarya R. As a student, I would like to know about the application and specific job opportunities in this.
Yeah, in India, a lot of people are working on bioinformatics. So those are software, not essentially they have developed, but they adopt the software from various public domain enterprises, and then they work on it. But there are companies like Schrodinger and other AI-based drug discovery companies that are coming up in the various countries. There are all lots of openings there, and I think it is a growing area. And this, if one focuses on just on genomics at the moment, there could be some limitations. But if you just broaden up a little bit and work on AI DDD, the chances are the openings are much more in that. Sorry, I have to turn off the generator and turn on the power. Just give me 1 second for me.
Okay.
Do we have more questions, Akhil?
Yes, ma'am. 2 more questions from YouTube streaming, and 2 more questions are being posted in the chat box. I'll ask them.
Yeah, please go ahead.
Yes, sir. Sir, if we, as we know that we are streaming in the YouTube also, there is 2 questions from YouTube live streaming, sir. Live chat. How can one visualize the dynamic interactions of RNA and proteins in lieu of fractal genome, especially the unbinding and structural changes included by this interaction for enabling genome function?
Yeah, as I said, this Craig, Francis Collins made a comment that our understanding of regulation is frighteningly unsophisticated because of the fact that the cancer itself is a genome misregulation disease. Therefore, Dr. Prash knows very well. He is working on long non-coding RNA and non-coding RNA and pvRNA, piRNA. You talk about the miRNA and circRNA, which is a love for me actually. So there are lots of these RNAs. Orchestra is going on. Whether it is actually in the tumor microenvironment or tumor immune microenvironment, or even if you're talking about normal genome, there are lots of regulatory finesse is happening with these RNA. Therefore, the coiled structure of the DNA with the histones and with the, I would say, HDACs and what they call the histone acetylation, and then you have methylation in the epigenome. All of these things are one complex biology, and we don't understand it yet to perfection. So I will reserve my comment on that actually. So.
Thank you, sir. One more question from YouTube live streaming, live chat. Why not choose a model organism with a small genome size, which will significantly reduce computation cost and also can be used to induce fractal defects and also test methodologies to correct them?
Yeah, this is a very good question. Remember, I was showing you the ZipF Mandelbrot that was done with a small mycobacterial genitalia kind of organism, and it could be done with a laptop computer, and you can show this kind of parabolic curve. And when you need, it is very important to understand, and I think you rightly pointed out, high-end computation is required for you to process huge amounts of data. And consider the fact that you have the level of data from one human genome, and then you have genome after genome after genome you are processing. Therefore, there can be model organisms by which you learn something, but until we get to the diseased, for example, cancer situation or any other disease for that situation, we will be able to address unmet therapeutic need. But in the case of a model organism, we'll be able to understand the biology. So I think the question is correct, but I think it depends on what you want to do.
Thank you, sir. There's a question from the chat box again. Can engineering students work in this field of research? I think you've answered this.
As the first point I addressed engineering people, I can point out to John Doyle of Caltech Pasadena. He is controlled dynamical systems, his research. He has done so much of work on genomics. So engineering faculty is important. In fact, in bioinformatics, we borrow signal processing. This is called genomic signal processing. We borrow from engineering's controlled dynamical systems, the concepts there. Then I told you about systemic, cybernetics, and informatics. So, of course, engineering skills are a boon rather than a bane for your informatics approaches. And imagine, I am not actually from the engineering side, or I am not from the informatics side. I had to go through a lot of learning. Being a domain expert, I actually study more biology than these concepts, but then I had to apply because I have to understand the mathematics underpinnings before I can come into this kind of concepts. Sure. So.
Yes, sir. So can fresh PhD graduates apply to pharma companies to work in the same field? If yes, what would be the best resources or papers to learn from?
Yeah, as far as the genomics, I will give it to you. This is being practiced by a lot of pharma companies, and you will have ample opportunity to work in target discovery, target identification, target validation, then drug development, and even in the case of clinical development, you can get involved in patient stratification, then studies involved with the bioinformatics and computational biology. So this is an open field for anybody. In fact, pharma companies are working on it. So if you ask me how many pharma companies within India working on it, I wouldn't say many, but there are some who are involved in it. But I really don't know who are involved at the moment with the deeper pockets at the moment in this area. But definitely when you consider companies of international caliber, they are all involved in it. They have a huge data science team in both in clinical trial sections and as well as in the drug discovery sections.
So you have answered one more question from Aishwarya through your last question, sir. But there is one more thing she has asked. Do we have students for internship? There is any companies or things, opportunities for doing internships or gaining experience?
Yeah, so for primers of the students and internships and all, you can approach the local companies. Many companies are doing genomics services and genomics products and things like that. So that will be a beginning point, stepping stone. And once you have some kind of good grasp on how to use those software for analysis, and you are a domain expert from the biology side, or if you are a computer science side, both ways it will be very helpful to get your hands on it. If you don't get your hands on it, you probably don't know much about how to analyze those sequence data and structured data. So now it is becoming very clear, both structure and as well as sequence are pretty important for drug discovery and development.
And one last question, sir. Could you please tell me which pharma companies are utilizing fractal analysis at the moment for drug discovery?
None, but I can tell you there is a company by name Foundation Medicine where they are working on it, on fractal work, and it is based out of US. And there is a company that my collaborator is having. It is called Holgen Tech. They actually started, but he is aged, and so he is not very active at the moment. He is above 80 now, so he is actually retiring. But other people that I can cite are involved more in diagnostic sector rather than in the therapeutic sector at the moment.
Thank you, sir. Thank you for your wonderful question and answer session. You have answered excellent with all the queries and cleared all the doubts of the participant. Thank you so much, sir. Now, I request Chair, CSI Chennai Chapter, Dr. K. Komathy ma'am, to propose the formal vote of thanks.
Yeah, before that, thank you, Dr. Akila. Before that, if any participant would like to directly ask him question, any query, please unmute yourself and you can ask. Dear participant, if anyone would like to ask the question directly to the speaker, please unmute and then ask. Okay. So no.
Good evening, sir. Just one question.
Oh, yeah.
So in the next 20-30 years, when this technology has reached its maturation and it has become a major player, how the ethics will be a huge issue now, sir. So what steps are being taken now to ensure that this technology is ethical? Because we are working with vast amounts of data and it's with people's lives also.
Yeah, that's a good question.
Very good
you might
question.
have heard about you might have heard about 23andMe, and you might have heard about other companies which are have to follow the regulation, of the FDA or any other lab-developed test, which is called LDT, lab-developed test. You can have the data actually, but you need a consent from the particular patient that you can release the data. You can't actually play with it's like a personal property of that particular piece of data is a personal property of the patient. So you need a consent. Definitely, these are things worked out, and there are ethical issues. Sometimes people can flout it, but essentially, what I mean is the data is not free for you to do whatever you want. You need the consent.
So in fact, for the cancer also, the first the data was used without permission, it looks like.
Yeah, this is a problem. I mean, the regulation is getting tight now because they say LDT, which is a lab-developed test, get it approved, and companies like 23andMe, they are not doing this kind of work. And the companies which are come up in like a Gordon 360 or Delphi or Exact Sciences or Foundation Medicine, they can test for the diagnostic test, but the data is something that you will not be able to use very liberally. I can tell you for sure. You need some kind of consent for that.
So
Yeah, there would be an ethical committee, no, sir. There would be an ethical committee for this sort of
that's not being worked out. I mean, in India, it is not very clear because we don't have strict regulations on this. But I think the regulations are just right around the corner because I think there are lots of companies working on it. And also, some of this, I am not very favoring that we should send Indian samples abroad. I'm not in favor of that. So I am for the cutting the cost. If you are sending your samples outside of the country, then it becomes you are revealing something. So our 4,000 to 5,000 sex of populations are there in India. It is a gold mine for any pharmaceutical company. Therefore, I'm sorry to tell you, we are at the just very nascent stages of this particular science. At least in our own country, it has got definitely a future for next 20-30 years.
Okay.
But what about the synthetic data? Will it sort of,
Yeah, that is one thing. People try to make synthetic genomes, cut, keep on cutting, and see how much I can cut the genome, and I can work with it, minimal genome. And these are all things, ethical considerations are there. Because now you can't do CRISPR-Cas9 to anybody because you are regulated. You are regulated because you can't keep on editing. Because the problem is you are editing at a place and you don't know that there are some cis and trans effects. What I meant by cis and trans effects is nearest neighbor sequence. And if there are some proteins that are getting affected. So thus such things are still being deliberated. It is not very clear even in a scientist's mind actually. But whatever they've approved, CRISPR Therapeutics or any other company which have approved, it's all for not complex diseases like cancer or multi-gene diseases.
Thank you, sir.
So genetic medicine and cytotherapy, the cell therapy are the future.
But this, what about this STEM technology? Huh?
Yeah, stem cell also is a future, but it is still nascent at the moment. Stem cell technologies. Yeah, and so I am also attempting something very in an ambitious way and finding funding for such kind of unproven in human kind of technologies are very difficult. And slowly we have to entice the people to put money into this kind of enterprise.
I think we must make Google, Microsoft, Meta to invest.
Yeah, large companies who are looking at it, Informatics, and we approached a lot of companies. In fact, at that time, our collaborators have been very vibrantly involved, and they even reached out to large companies in the US. But right now, they are working on some Hungarian establishments and things like that. So all these, my collaborators are a little bit aged, anywhere between 75 to 82. So they have come to a level of retirement now. So I think it is better to take from some youngsters and work with them at the moment.
Excellent.
Nice. Good questions. Thanks. Thoughtful questions. Thanks a lot.
Very interactive session.
Yeah. So thank you. Good evening all. This is Dr. Komathy, Chair, CSI Chennai. On the outset, I thank the organizing units, namely IEEE CS Madras, CSI Chennai, ACM Chennai, and Bioclues for arranging this webinar today with the topic Programmable Biology, Fractal Genomics, and Precision Medicine. Informatics to Rescue. We perceive that the new era of computer-enabled biotechnology companies that integrate machine learning, deep neural networks, and artificial intelligence to not only predict the safety and efficacy of new drugs, but discover, design, and develop better therapies for patients. The webinar has focused on how genomic approach improves the management of critical ailments through precision and personalized medicine. We sincerely thank Mr. Ramanujam, founder and chief science future officer, Prodigious, Quant-Epigen, Bengaluru, for his estimable presentation on computational biology and informatics in fractal genomics and precision medicine. He introduced how mathematics, especially the fractal geometry, infiltrated in biology and genomics, where fractal refers to a pattern that repeats forever. Genome is a fractal. For example, oncological mathematics is a multidisciplinary field that uses mathematical models and simulations to understand, predict, and describe cancer. Mathematical oncology can help us understand how cancer starts, how it progresses, and how it responds to treatment. He illustrated how fractal defect is neutralized using genome-guided therapy, which is vital for prognosis with precision, accuracy, and efficiency. The technique of genome editing leads to the emergence of a new informatics-based industry and genome engineering. Genome editing is currently in its early stages waiting for approval and recognition. I thank once again Mr. Ramanujam for clarifying the question raised by the participants offline and online as well. I thank Dr. Prasant, founding member Bioclues, for his presence and supporting this webinar. And sincerely thank our mentor, Mr. H.R. Mohan, Chair Events, IEEE Madras, who worked diligently to bring speakers covering the multifacets of computing technologies for the benefit of our members. I'm thankful to the participants who joined us in live Google Meet and YouTube live streaming. I thank the event team members from the organizing units for their support in arranging this event. With these warm words, we move to the end of today's event. Thank you all.
Thank you very much. I appreciate it.
Yeah, thank you.
Thank you. Thank you, sir. Thank you for the wonderful session.
Thanks a lot. I appreciate it. Thanks a lot.
Thank you, sir.
Good night, sir. Thank you and good night. Yeah, yes, no, yes.
I think lecture Q&A was longer. Looks
Yeah, yes. அது ஃபைனலா அப்படிதான் போகும் சார்.
Because the topic is of some sort of a different topic.
I think biology